Today we're going to finish, I think, chromosome structure. Mostly what we're doing today is getting to the answers of a few questions that have been raised the last couple of classes. So I do intend to work fully through some more examples and answer your questions. All right. So just so that you have answers to these questions, what was the enzyme that cuts this site? This is not important to remember, for you to memorize that is. I just want to make sure everyone's got the answer. Not one. And those numbers are always Roman numerals for who knows what reason, but not 1, not 2, not 3, etc. Okay, what we didn't get to last class, and I do definitely want to be able to answer, what mutation would cause AGG CTT to become a HindD3 restriction site? So, don't answer yet. I just want to get a show of hands. I want people to raise hands, please, to answer this question. What's the first step towards solving this problem? Don't say it out loud. Just raise your hand if you know the answer. What's the first thing you need to do if you're going to make that a HindD3 recognition sequence? What's the first piece of information you need to know? Yes?
The sequence that is HindD3.
Okay, so you want to know what is a HindD3 restriction sequence. So you need to find that enzyme and what sequence it recognizes. What is the sequence that it recognizes?
Wouldn't the second G become an A?
What's its restriction site for HindD3? What sequence does it recognize? Should we Google it? AAGCTT. So we start with AGGCTT, and this is the HindD3 restriction site, which you did, presumably, like you said, by typing HindD3 into a search bar on a browser, and the first hit probably says something like, New England Biolabs, this is HindD3. That's the enzyme that recognizes AAGCTT. So the only change we need to make to turn the original in black into the restriction site, as you said, was to mutate that G to a C, to an A. That is, mutate, a single nucleotide change. Change one letter to another letter, mutation. So if you were an organism and you had AGGCTT, the top sequence, in your genome, somewhere on a chromosome, the enzyme HindD3 would not be able to bind to it and cut it. But if you made this single change from a G to an A, then you've created a HindD3 restriction site, a DNA sequence that the enzyme will recognize and cut. And these restriction enzymes are things that molecular geneticists use all the time in the laboratory to clone, to take different pieces of DNA from different organisms and stitch them together. It involves cutting apart the chromosomes of an organism that you want to get a gene out of, say, and then cleaving the chromosome of a donor. Let's draw this out so it's a little bit more explicit. So let's say you've got organism A and organism B, and organism B has a chromosome, and organism A has a chromosome, and organism A has a particular gene. Nomenclature, this is something that every geneticist recognizes, so it's really important to pay attention right now. The gene that I'm interested in is YFG. That stands for your favorite gene. It's the gene name of choice for every geneticist when they're talking about a generic gene. Your favorite gene, you make it whatever you want. It's a generic gene. So I don't know what it does, but let's say you wanted to take the YFG gene out of organism A and put it into organism B. One of the ways to do this is to find a restriction enzyme. I don't know, let's say there's an EcoR1 site on both sides of your favorite gene. So you could extract the DNA, you could take the DNA out of organism A, apply EcoR1, and EcoR1 cuts the chromosome everywhere there's an EcoR1 site. And so you isolate that gene, and you can put it into the chromosome of the other species. But there's something you have to do to integrate this section of double-stranded DNA, this part of a chromosome, into organism B. You have to use an enzyme again. So what do you do to the chromosome of organism B?
Would you use EcoR1 again to find the same set of nucleotides to put it in?
You could. It could be the same, or it could be a different enzyme. So it might be that there's a specific spot on this chromosome you want to insert your trans gene, your gene from organism A. So you might cut it there with HindD3. And what that does is that separates the chromosome into two parts, and that gives you a spot in the middle of the chromosome to insert that transgene. If you're cloning. This is one type of cloning. This is molecular cloning. This is stitching together pieces of DNA from different organisms. That's different from organismal cloning, like cloning humans or cloning sheep or cloning cats. One word, two totally different meanings. This is molecular cloning. Recombinant DNA. Taking DNA from different organisms and putting them together. Molecular cloning. It's not like making a different version of me. That would be a different type of cloning. So this illustration is just to give you an idea of how scientists use restriction enzymes. I'm not going into details because I'm not necessarily going to ask you about details on exams. Just the concept that you use enzymes to cut pieces of DNA and you can move them around. All right. So I want to show you a few of the submissions of this exercise I asked you to do for today. I was able to post responses to some of you who turned this in. This is not for points, just for me being able to give you feedback. So if I haven't made it clear yet already, this class structure, this is one of the main ways you get feedback is if you turn in the assignments on Google Classroom on time or early enough, I'll look at them and sometimes I will randomly pick yours to anonymously show in front of the class, and then as a class we'll go through analyzing the response. So I won't necessarily address, I don't have time to address everybody's response, but hopefully you'll see something here that helps you self-evaluate how you answered this question. And if you have additional questions, then I do implore you to come see me after class in office hours and so forth. So this is one of your submissions. I'm not sure whose, it's anonymous. Find restriction sites was the goal. So let's take a minute, look at the 3 sites that this student underlined. They said they thought these were restriction sites, and I'd like you to tell me, in about a minute, we're going to vote on which of these are or are not restriction sites. Basically, are they palindromes? And I'm happy to hear the whispering. That's good. Which one makes you think the hardest about whether or not it's a palindrome? 1, 2, or 3?
C,
2. Okay, so is 1 a palindrome? Does it read the same from 5' to 3' on both strands?
Yes.
GATC. And number 3 also reads the same 5' to 3' on the top strand as it does on the bottom strand, GGCC. So what's weird about number 2? Is it a palindrome?
Yeah, it is a palindrome, but the nucleotides aren't correct.
And this is totally not whoever's fault. There are actually multiple submissions that look like this, which is why I'm using this as an example, because I didn't tell you this last class. So this is a great example. Yeah, for a palindrome in genetics, all of the letters in the palindrome have to be base paired with all of the other letters on the other strand that are part of the palindrome. In other words, you can't have offset edges to the palindrome. They all have to be right on top of each other, the whole palindrome. Like example 3 and example 1. So yes, it's a palindrome, but no, it wouldn't be a restriction site because those enzymes will only bind palindromes when the whole palindrome is base paired with itself, like 1 and 3. So simple misunderstanding and my fault because I didn't specify that detail last class. Okay. Now, I showed this one not to evaluate it per se, but to make a point. What do you notice up here? What are some things that might surprise you based on how you answered this question? Is there anything notable? I mean, I asked people to identify restriction sites. Most students turned in something that had one restriction site underlined. How many restriction sites are there in this short piece of DNA?
More than one.
It's a lot. Yeah.
Or they're extended past the edges of where they underline.
Yeah, so 2 points. Restriction sites can overlap. So that's 2 palindromes. The underlined part and the highlighted part are 2 different restriction sites. They just overlap. 2 enzymes couldn't bind there at the same time, but either of those enzymes could bind there individually. That happens again up here. GATC is a palindrome, but so is TCGA. That's fine. Some palindromes are larger than others, 4 nucleotides, 6 nucleotides. And then, likewise, that restriction site has 2 palindromes embedded in, 1 palindrome embedded in a bigger palindrome. So if you got that, for example, on an exam and I said, what restriction enzyme binds there? There are actually 2 different choices. There are enzymes that will only bind GATC, but there are others that will bind GGATCC. Multiple answers. Both of them would be right. So GATC in itself is a palindrome, but so is GGATCC. That's fine. Any concerns about any of this? Questions, clarifications needed? Palindromes, yeah.
Should we memorize the enzymes that go with which palindromes?
Please do not memorize enzymes and palindrome sequences. That's a great question. I'm glad you asked. Yeah, the point of this, let's get back to, I hate memorizing things, especially when you've got digital devices. You can look this stuff up if you need to. So don't memorize it. Everything I'm doing here is for example. You don't need to know Hindi 3 and its recognition site, EcoR1 and its recognition site. Practicing molecular biologists and geneticists do, but that's just because they use them so frequently that they remember it. You're not at that point in your careers yet, unless you work, for example, in Dr. Yoon's lab, where they do a lot of molecular cloning of bacteria, and you might actually use these pretty frequently. Okay, what about GGCCC? Is that a palindrome?
No.
So how do we edit it to become a palindrome?
Just take the last.
Yeah, just GGCC, yeah, that's a palindrome. So what's an interesting feature of these palindromes? This one broke the rule. A nucleic acid palindrome will always be some number of letters long.
Oh, even.
6 or 4 or 2 or 8, right. So a palindrome is always an even number of letters. Or at least almost always. You can't make a palindrome out of an odd number of letters. The other thing that I should mention, although I didn't put this on a slide, is that some people, for example, let me just write it up here, claimed that AT in the context of the rest of the chromosome is a palindrome. Is it? Yeah. But technically, there aren't any restriction enzymes that recognize a 2-base pair palindrome. The smallest restriction enzyme recognizes, or the restriction enzyme that recognizes the shortest sequence is 4 nucleotides. So yes, technically that is a palindrome, and you would have gotten full points on that if that had been on in some sort of an assignment. But again, I didn't tell you. And why do you think the reason is that there are no restriction enzymes that recognize 2 nucleotides? Anybody? What do you think?
You find it all over the place.
It's so common.
DNA, endonucleases.
Right. The frequency of AT, just those two nucleotides next to each other, is so common in DNA that if you threw a restriction enzyme on there, it would chew up the whole chromosome. There'd be nothing left. And what I haven't told you yet is the reason why restriction enzymes exist. Remind me some other day and I will tell you, because that might make a lot more sense. But bacteria evolved restriction enzymes to do a specific job for bacteria, and that is the reason why it's important that they not cut up everything everywhere. Okay, so time for a little exercise on your part. I'm going to launch a Google Classroom question. And what I'm asking you to do is, I'm going to ask you to go to PubMed and find a manuscript that contains a karyogram. Any species, doesn't matter. And then when you are at PubMed, in the browser window, copy-paste that URL to that paper, the record on PubMed, and post that to this question that I'm about to post to Google Classroom. We've got like 5 minutes for this. And of course, this is not being scored. It's just for practice, looking up papers on PubMed. Plus, I'm going to hopefully use one of your responses to discuss karyograms. A little bit more practice, since a lot of you asked for more practice, karyograms, karyotypes, ideograms, and so forth. If you have a question about this or any concerns, please raise your hand and I'll come around and help. Okay, for those of you that already turned in an answer, and if you haven't yet and you want to, keep working on it. This is practice, looking up manuscripts on PubMed. What did you do? Go to the PubMed website. What search term did you use? Karyogram. Okay, fair enough. That's what I would do. And it looks like everybody is putting in a PubMed URL, as I asked, so that's promising. So when I do that search for karyogram, this is what I see on NCBI. And a lot of you happen to pick the first one, which is totally fine. So looking through the URL, almost all the URLs look the same. And it's to this study. So the one point I want to make on this slide is really straightforward. If you're ever trying to do research on something and you want something that you can look at immediately and not have to pay for, just look for that free PMC article there under the record. That'll tell you that when you hit that URL, it'll take you to a web page where you can get the actual full text of the paper that you're looking for. So free article, free PMC article, it all means the same thing. So if you open that, then you'll see links on the upper right here. So this paper, what journal was this paper published in? The citation's up there on the top, just above the title. That's the name of the journal. It's the abbreviation of the journal. It's called Peer Journal. And that's why one of these full text links is to that journal's website. So you go directly to where the article was published and get the free full text of this paper. This link is only important for you to know as taxpayers, if you are, because all federal research that's done by the U.S. Department of Agriculture, National Forest Service, National Institutes of Health, anybody like that, National Science Foundation, if we receive federal dollars to do the research, then we have to make it publicly available to the people that paid for it, which are the taxpayers. So every paper that is publicly funded will have that. It's the PubMed Central. This website has to host a free version of all papers that are funded by federal tax dollars.
What if people from not America wanted to access it?
Well, so people from other countries can still get to this website and see the free versions. What doesn't work is people that are funded by other countries that publish with non-US taxpayer dollars funding don't have to make their papers publicly accessible. Some countries do, some countries don't. And generally the field is trending towards, we want to make all the knowledge freely accessible. I mean, that's kind of why a lot of us got into science, was to find things and then tell people the cool things we found. So having to pay for subscriptions to journals is sort of a necessary evil at this point. But not every paper you may have already discovered is freely accessible. So if I open this and look at the full text, then presumably at some point I'm actually going to see a karyogram. Ah, excellent. So just briefly, what are we looking at?
On each one on the right is a it's just a spread.
Yep, metaphase chromosome spreads.
And then on the left is the karyogram.
Karyograms, where they've cut out the individual chromosomes, rotated them, and then lined them up based on size, left to right, biggest to smallest, and centricity, presumably. And we're going to talk in just a second about an exercise to practice your chromosome centricities one more time. What's the difference between the left columns and the right columns? These are the species, so same species, different sexes. So do you see any differences between the sexes? If you compare left to right, left to right, left to right. Any differences? Chromosome number, chromosome size, they all look pretty much the same.
I think the ones on the right have chunkier chromosomes.
Chunkier chromosomes?
They look chunkier.
Yeah, that's probably an experimental artifact. Differences in how thick the chromosome arms look, or how well separated the arms are, the sister chromatids, is
Maybe a little bit longer.
artificial. Yeah.
A little bit.
We're definitely going to get into how you prepare these slides at some point during the semester. And then we're going to see whether or not the relative lengths of the chromosomes has anything to do with anything. The short answer is no. So thanks for doing that. All right. Now, a large cry came up from the audience a couple of classes ago when I said, what do you need to know more about? What do you want more practice on? And it was from about half of the audience. What the hell this N, 2N, 4N thing is. So we're going to go through it one more time in detail to make sure everyone's on the same page on karyotypes. So this is a mouse. Karyogram. Think Instagram. Instagram's pictures, karyogram pictures. Okay. It's the karyotype that goes up there in the corner, the numerical description of what you see in the Instagram. So the first two things you should be able to get out of looking at any karyogram is, what's the ploidy? So how do you figure out the ploidy again? That's the number of copies of an individual chromosome. So here we have 2 copies of chromosome 1, 2 things that are labeled 1, thing 1 and thing 2. So that means that this is a 2-ploid, diploid individual. So that's how you figure out ploidy. You look at a karyogram and you say, okay, how many versions, how many copies of each chromosome are there? If it's 2, it's diploidy. If it's 3, it's triploidy. If it's 4, it's tetraploidy, but I would accept quadruploidy. It's tetraploidy. Yeah. Okay. Now, how do you figure out the other highlighted thing? The N. What does N mean? Number of what? It's the number of different chromosomes. So how many different chromosomes are up here? Yeah, exactly. 19 plus. So it's there's chromosome 1, chromosome 2, chromosome 3, chromosome 18, chromosome 19, and then there's a 20th pair, which are the sex chromosomes. So that's N, the number of different chromosomes in the karyogram. Yeah?
Is having one of each would be haploid?
Yes.
Okay.
Yes. So yeah, exactly. That's the unfortunately termed one. It's not unoploidy or monoploidy. Unploidy. Yeah. So having one copy of each chromosome is haploidy. And where do we see that in humans and many organisms? There are haploid cells
Oh, gametes.
that are gametes. Our sperm and our eggs are haploid. They have one copy of each chromosome. And that's why fertilization is necessary, because that's how we get back to diploidy. Half of our chromosomes, 23, come from the sperm, another 23 come from the egg. And together that produces a single cell embryo that has 46 chromosomes, two of each. 2n equals 46. So this is n, 23, n, 23. And that's 2n. Diploid organism, 46 chromosomes. So that's how you do the math. So if you want to do something like 2n equals something, humans have, what's our n in humans again? 23. And we're diploid. So if we're diploid, we've got 2. Our n is the number of individual types of chromosomes, 1 through 22, and then the sex chromosomes. So you just fill that into the formula. 2 times n, 23, equals 46. So that's where that 2n equals comes in. 2 is from the Ploidy. So that could be different depending on what species you're looking at. Could be, what did we say? Some plants, salmon, are tetraploid. They have 4 copies of each chromosome. Talking about a salmon, it would be 4n equals something, would be its karyotype. And that n, we would have to figure out the number looking at the karyotype. How many different chromosomes does it have? And then do the math. 4 times n equals whatever. So if it was a salmon with, I don't know, 6 different chromosomes, then you'd expect to see 24 things in a karyogram, 24 chromosomes. Now, I know some of you already know the answer to this because we talked about it before class. Here's another human karyogram. So what's the difference between this and a normal human karyogram, and what's its ploidy? So think about that for 15 seconds. And I'm going to hope somebody from the, I don't know, middle here of the class. Wait for one of you in the middle of the class. Yeah, that also includes you, sir. What is the ploidy of this karyogram? How many copies of each single chromosome are there? This is chromosome 1. There's 1, 2, 3, 4 chromosome 1s. So that's 4 ploidy, which we said is tetraploidy or quadruploidy. Tetraploidy. This is a tetraploid human karyogram. That's not normal for humans. This is lethal. You can't survive as a human with 4 copies of every chromosome. Where do you think this came? I don't remember where this came from, but I have a pretty good guess.
My guess would be that the gametes did not divide correctly, so one daughter cell got all of the chromosomes and the other got none, which would be weird.
I'm going to Google this really quickly so I can try to remember. My guess is that this is from a cancer cell. And the reason I suspect that is because a lot of cancer cells go crazy when they do DNA replication and cell division, and sometimes you wind up with cells that are tetraploid, octaploid in a human. Octaploid, 8 copies of every chromosome. That's, if I remember correctly, that's where this came from, was a cancer cell line. I might be wrong. If I am wrong, please tell me when you figure out where this actually came from. But this is just an example of tetraploid. Let me see. Okay. Let me start a Socratic activity. So if you'd all please log in. And then we'll end with a couple other exercises. This is from another one of your submissions from the NCBI exercise. Just two, quick little practices for centricity, because some people ask for more practice on that as well. So just number the chromosomes 1 through 5, and then take 1 minute, see where you place the centricities. And by the way, since I never mentioned it explicitly, in ideograms, the little point where the 2 chromosomes look like they pinch together is how the centromere is identified in an ideogram, in the cartoon. Which one's metacentric? Centromere is in the middle. 3. Each of the arms of the chromosome are about equally as long. The centromere is in the middle, that's metacentric. M, metacentric, middle center. Submetacentric, slightly off, would be which one? Number 2 is a submetacentric. The centromere is a little bit lower than the middle. Do we have an acrocentric?
5.
Okay. Which one is number 1?
Telocentric.
Number 1 is telocentric. It doesn't even look like it's got a centromere, and that's because the centromere is at the very end of the chromosome, which means that 5 is, it doesn't mean that. 5 is acrocentric, which means there's such a tiny little small arm that you can barely see it. It's there, but it's just a little dot. What didn't we do?
4.
Oh, what's 4? Also metacentric. And I know this is in the video, but this is also in the list of tasks or points for this chapter. So, short versus long arm. This is one you just have to remember. The cytogeneticists don't call them just short arms and long arms when there's a submetacentric. They have specific terms for them. Do you remember what letters they use to designate the 2 arm lengths? The short arm is P, the short arm, P arm, and the long arm of a submetacentric is called the Q arm. So mind your P's and Q's, as they say. Okay, last one of these, and then I'm going to end with a little story about us and our closest relatives. So here are not ideograms, but chromosomes. So what are the centricities? We've got chromosome 1, 2, and 3. What's number 1? 1 is certainly metacentric. How do you feel about number 3? Not how do you feel, sorry. What is the centricity of number 3? What do you feel that the centricity of number 3 is? Does anybody disagree? What else might it be? If it's not telo, it's definitely acro. So I will accept either answer as long as you can explain why you chose either. I kind of tend to think that maybe that's the centromere there. That's where the chromosomes are the narrowest, which would mean that there's something up there, which is a short arm, so that might be an acrocentric, not a telocentric, but it's definitely open for debate. And then what would number 2 be? Either submetacentric, acrocentric. Again, as long as you can explain why you chose that answer, I would accept either. Again, there is a formal definition of how you calculate the centricities that I showed a couple of days ago. We're not going to use that. I just wanted you to know there is a quantitative definition of how long the arms have to be. But for that, for number 2, yeah, that could be submetacentric or acrocentric. Submetacentric or acrocentric. So, to close, 2 stories actually. This one is about the question, how do you make a karyogram from a metaphase chromosome spread? And this is one that I made when I was in grad school, so I can show you the before and the after. This was the nucleus of a 3-spine stickleback fish that I was looking at, chromosomes affixed to a glass slide. And the easiest way to tell what happens, I think, is to focus on the weirdest looking chromosome, or at least the most unique looking chromosome, which I think is the one in the upper left. Kind of has 2 skinny arms that go all over the place. It doesn't look like what we think of as a chromosome. So you'll see in the next slide, I cut all these apart, rotated them so that they could line up next to each other, and then organized left to right, biggest to smallest, into 4 different classes of chromosomes, plus the sex chromosome. So where does that weird chromosome wind up here? The one with the really weird arms. Yeah. It's one of the submetacentric chromosomes. So did I actually know which of these were supposed to be paired with each other? A lot of these are the similar shape and size, and these are not banded. So I don't really know for sure that when I published this paper that I actually got all of the homologous chromosomes really paired up next to each other. It looks good though, so it suffices. It's enough to prove at least how many chromosomes this species had, which was the point of this study. Okay, finally. Do I have enough time? Oh, yeah. Our closest relative is the chimpanzee. We're all monkeys except we're special. So the story I'm going to tell you about is about our 3 closest relatives, orangutans, gorillas, chimpanzees, and then us. All primates. We can talk and do some other stuff. So these are not actually karyograms, are they? What are these? My bad. Primate ideograms. So, left to right, up here in the upper right-hand corner, human, chimpanzee, gorilla, and orangutan. So each of these groups of 4 are those 4 species left to right. This is the human version of chromosome 1 and what it looks like when it's banded. Chimp, gorilla, and orangutan. Do you see any differences? Where do you see differences? What sort of differences do you see? Ignore the colored dots. Our which one?
The first one.
So our chromosome 1's a tiny bit longer. Other differences. There's slight variations in length. What about banding patterns?
Number 4, the
where
Oh,
the
number 4.
banding patterns are different, and also where the little pinch is, yeah.
Yeah, so the centromere location changes from human to chimp to gorilla to orangutan a little bit. Most of the banding patterns are the same, but there are some minor differences. So the point of this is a paper from some cytogeneticists about almost 2 decades ago now, 2002. Basically, it shows how genetically closely related we are to our closest ancestors. Not only do we have very similar DNA sequences, but then not surprisingly, because the DNA sequences of our chromosomes are so similar, the chromosomes themselves also look and band in similar ways. There are some differences, though. Chromosome 3 is a big one. Orangutans have their centromere in a much different spot than humans, chimpanzees, and gorillas. And we're going to talk later in the semester about large-scale chromosome rearrangements, translocations, inversions, and deletions, and how this sort of diversity arises. The one chromosome that wasn't shown on that ideogram, though, was chromosome 2. And that's because chromosome 2 is the one big difference on a chromosome-wide scale between humans and the non-human primates. And that's because in chimpanzees, gorillas, and orangutans, each of them have 2 different chromosomes. And in the ancestor to just us, in the humans, in the Homo sapiens that gave rise to all of us, those 2 chromosomes fused. You could imagine the chimpanzee, these 2 chromosomes sticking together, and that makes what humans have as our chromosome 2. There's one major, then, karyotypic difference between humans and the non-human primates, and that's that we've got the same types of genes, they're in the same locations, it's just these 2 chromosomes stuck together and became a single chromosome. That means that in the chimps, gorillas, and orangutans, these chromosomes are referred to by humans as chromosomes 2A and 2B.
So in the orangutan chromosome, and then also on the last slide number 13, 14, and 15, had what kind of looks like either a piece floating off or like a little halo.
That's a satellite DNA. It's part of the chromosome, but it doesn't contain any genes, so they just sort of draw it like that. Yeah. What's being referred to is sometimes on an ideogram, you'll see a little blob down there that's just below or just above the chromosome. That usually represents on an acrocentric chromosome the tiny little bit of chromosome that's on the other side of the centromere. Okay, so I think at this point we've covered all the vocabulary, we've done all the tasks, and we've answered the question, why do chromosomes have stripes, perhaps repeatedly. And for next class, then, we are moving on to the next chapter. So this is an appropriate time, if you haven't done it already, to, at the end of this chapter's 3 or 4 pages of the PDF, there are those fail exercises. First attempt in learning, fail. The exercises that I'd like you to try to do before we get together and start talking about the next chapter. So please make sure you've tried those. Read the next chapter, 1.3, it's not so long. There are a couple of sections of textbooks to read, a few videos, but not long in terms of minutes. And try just the first exercise. There are a lot of exercises in the next chapter. Just aim for the first one. We'll talk about that on Wednesday. Have fantastic weekends.